So, I do know people get really stressed by presenting. Um, you are not being graded on like being super suave up there. You're just being graded on, are you actually kind of sharing the information? You know, so you do have to present. Some people asked, like, sent a message like, I'm nervous presenting. Is there another way to do this? And it's like, no, I would like you to present in person and kind of talk about, and that way you can also, people, it's nice to see people interested in your stuff. It's nice to be able to respond to people's questions. So, um, but know that you are being, this is where you're graded more on just like, showing up and participating and doing it rather than kind of, are you, this is not Toastmasters, right? You are not being graded on being like an accomplished public speaker or something. Um, so if your voice quakes or something, you're not losing any points, right? So don't worry about that. Um, it's also, it's nice as the audience members to just, give full attention to whoever is currently talking. It's especially, especially on Zoom, it's easy to like be like, doing your Christmas shopping, or doing, or else prepping for your own talk that you're all nervous about to come up. I would request that, just out of kind of respect for the presenters, is like, give them your attention so they feel like they're being heard. you know and I will say that, looking at these, I think you'll be interested in what people have to say. People have covered a lot of cool stuff here. So that being said, we'll dive in. Um, and I have, like, Emily. Do you mind starting the whole thing?
Who?
I said Emily, but
Ah,
we could
My
do.
apologies. Sorry, it cut out. But sure, I'll go ahead and get this
And
over with.
do you want to share your screen, or do you want me to run your presentation?
I can attempt to share mine, and if it starts to get like if the connection starts to get too bad, just let me know.
Okay. So, you should have permission to share screen. So,
Uh,
and we should have. So let's give a hand for Emily, who's taken the plunge here and is the first person.
um, sorry, it's not showing it yet. One second. Okay, already having technical problems. Sorry, it's not letting me. There we go. Okay.
Oh, there we go.
Um, so we actually have already started to talk about this in class, and we just talked about it. So I've always been pretty interested in probiotics and how they've affected us, especially after taking my microbiology class about a year ago. So in my research that I was going through all these different research papers and everything, um, found out quite a bit more. So just kind of an overview. Sorry, I'll move the Zoom window out of the way. Just kind of an overview, like what probiotics are. We've already talked about it, but basically they're living organisms. They can be bacteria or yeast, and they can have uh, positive benefits to us. That's what classifies them as probiotics rather than like pathogenic microorganisms or disease causing. So you find them naturally in fermented foods. Just to list off a few, it's like kimchi. This huge thing right now of kombucha, but you can also find them in like your yogurt, and of course dietary supplements. So they are, of course, heavily in our gut and intestinal system, especially our large intestine. But you also find them in your skin, your nose, your mouth, lungs, and genitalia. And there are different genera or types of microorganisms. So some, again, are beneficial and others are harmful. So I came across this image in one of the articles that I came across, and I liked that it kind of gave a general summary of how you know what we eat can start to impact everything else. And so when there is that imbalance of our microbiome, which can occur from, again, things that we eat or environment, then we start to see this cascade of other things going on. So we have issues in our gut with inflammatory bowel disease. We have colonic carcinogenesis or colon cancer, infectious diarrhea, impaired immune systems, again, inflammation there. There's actually linking towards eczema and atopic dermatitis and skin hypersensitivity. Um, and has really been fascinating is seeing the link of our gut and brain and seeing that it can actually have effects on our on depression and anxiety. So again, just kind of part of the intro, just that our food can actually affect our microbiome. So this was a cool image, just clearly showing that actually having plant-based protein can, as you can see, increase certain kinds of bacteria like bifidobacterium and lactobacillus, whereas it reduces some of these other ones like Clostridium. And the two that it's reducing there are actually pathogenic microbes. Then the ones that it's promoting are ones that are actually beneficial to us and can help us. And as you can see, the linking there, it increases the gut barrier and increases short chain fatty acids. And then the opposite is happening when we're having animal protein. So we're seeing this increase of more pathogenic microbes, and it's now causing other issues and leading towards irritable bowel disease and heart disease. This was another great image that I came across. It's pretty extensive, but just to give you an idea of how many bacteria that we really have, it's really extensive. And then also seeing some specific changes that are going on physiologically and, how's that affecting us as far as disease. So there's some linking there of reduces abundance in obesity, increases abundance in irritable bowel disease, and then kind of more towards the bottom there, we see there's anti-inflammatory effects. So effects on psoriatic, my apologies, psoriatic arthritis. And also, of course, we're seeing potential of UTIs. Um, so just to focus more specifically on gastrointestinal health, there are specific strains that have actually been shown to alleviate the symptoms of IBD. So, lessening the abdominal discomfort, bloating, the urgencies, and flatulence. But also, as we've already mentioned in class, by having antibiotics, it's now wiping out some of these beneficial microbes and providing a more favorable environment for these pathogenic ones. So, you'll hear this a lot in healthcare about antibiotic associated diarrhea. And one of the big ones that you hear about is C. diff. Um, and this is, I'm sorry.
I'm sorry, I thought it was muted.
Oh, that's
I
okay.
apologize.
So this particular one can definitely wreak havoc. Just personally, firsthand, I had worked in senior care, and anytime a patient had this, it was just like, oh my gosh, put that person on isolation, all these extra precautions because that things it spreads very easily, and it does wreak havoc in our gastrointestinal system. As you can see, it produces these exotoxins, toxin A and B, which triggers inflammation and this diuretic reaction, and it really can be a dangerous situation, especially for elderly or other immune compromised, because it can be very hard to manage, and it can take some more intense antibiotics to try and wipe it out because it's more resistant. So there is some evidence that has been supporting, though, by having these probiotics, it reduces your chance of the infections as well as the duration and reoccurrence. And I've had some patients in the past that have had several reoccurrences of C. diff because it's just persistent. And they end up having the fecal transplants, actually. So I definitely pushed that when I was working there, of having patients take probiotics. The other areas, of course, are on our skin, and even our vaginal health can be impacted. And again, just the fact that when there's a shift and change in our microbiome in these areas, we could actually see a link there, even between our gut and our skin health. So we become more hypersensitive. There's more chance of atopic skin conditions. Even in children, they did a study with mothers and provided them with antibiotics, and saw that actually significantly reduced their chance of their children's getting atopic eczema within their first 2 years of their life. And there's also the idea, in reintroducing these beneficial microbes are going to be helping that environment, and so it reduces the reoccurrence of vaginal infections. And again, a big theory right now is how having such a clean environment has now significantly reduced our exposure to these microbes. So that's actually been on my mind even more recently because of how much we are more isolated. We're staying more so indoors. We're using a bunch of hand sanitizer. We're washing everything and sterilizing everything as best we can. And so it's going to be really interesting to see how that's going to be affecting our next generations, even in like these kids that are growing up in this kind of environment, rather than playing outside and being in the dirt and being exposed. So we shall see how that plays out. And then in our immune system, it actually does, we were just talking about immunoglobulins. So there's potential that it increases IgA, which can have an effect on our humoral immune system response. And then how it's also protecting us is being a competitive inhibitor of pathogenic microbes. So making it more difficult for these harmful microbes to thrive because they're competing for the same nutrients and just space. So there's also the production of short-chain fatty acids, as I mentioned, which affect our immune system through the G protein coupled receptors and other epigenetic modulation. So that's actually affecting our gene expression. And they cause a decrease in secretion of the cytokines that we were just talking about. And probiotics help magnify our beneficial microflora. So again, just giving or providing more of these beneficial microbes. So as I was talking about, there's this whole gut-brain connection going on. So this was a cool image, just talking about how there's a two-directional pathway that exists between our gut and our brain. So by altering the gut, now there's idea that it's going to be altering our brain functions and our mood. And so there's some connection with the depression and anxiety. And a specific study that I came across, there was actually a significant improvement in their BDI scores, which is a measurement of basically that they use to measure depression in those that have major depressive disorders. And by putting these patients on an 8-week program for probiotics, there was actually significant improvement even than those that were on a placebo. And placebo effect can be very powerful in itself. So that was pretty impressive to see. And then even in animal studies, they have demonstrated improvement in anxiety, but so far there hasn't been quite as much research showing that in humans so far. So we definitely need more more research done there. But just kind of looking over this image, you can see that this effect between the gut and the brain, actually, there's some connection there with even schizophrenia. You can look through and see that there are specific microbiota modulation of our central nervous system and homeostasis. So, what are the mechanisms and things that are happening when that's affected? And just to kind of wrap it up, overall, probiotics are seen to be safe, especially more specifically the lactic acid producing bacteria. They are considered to be commensal microorganisms, so they don't necessarily cause harm to their host. They're just kind of hanging out. And most probiotics have even been classified to be generally recognized as safe. Some risks do occur, though, in some more rare cases, but even more specifically for those that are immune compromised. But there have been some incidences of them causing some infections as well. So just being especially careful for those that are immune compromised, especially. But there is some substantial evidence supporting probiotics for improvement of our gastrointestinal health, immune function, vaginal health, as well as some evidence for treatment in mood disorders. So there is still quite a bit of inconsistencies, and we really do need some further research to kind of pinpoint and determine the efficacy in using probiotics for the treatments and diseases and disorders, but it's pretty promising so far.
All right.
That's all I have.
Great. So now we can open it up to questions. Anybody have any questions for Emily?
Oh, I just wanted to make a comment. I thought it was really interesting about the plant protein being associated with I guess better gut or better microbes and microbiomes, and the animal protein actually was linked to cardiovascular disease, but like from a bacterial standpoint, I didn't really know the relationship before, so I thought that was interesting.
Yeah, thanks. I wanted to touch on it a bit more, but it just started to be like far lengthier paper. But basically, when they talk about prebiotics, it's just basically the nutrients for our probiotics, and so our plant-based foods can be prebiotic for that. So I think that's where there's a little bit more of the connection, or maybe the reasoning behind why plant-based is more beneficial for us.
Cool.
So, all right. So that was awesome. Any more questions for her?
I think it was super cool,
Thank you.
Emily. Yeah, super interesting.
All right. So thank you for that. And now back to Maria. Maybe you ready to go? I don't hear you. Maria?
Yes. Let me see if I can share my screen.
Again, if you don't, I can run your presentation as well.
Is that it?
Yep, you got it.
Okay. Well, hi, my name is Maria, and my paper was about magnesium and depression. And well, first, I want to give a little background why I chose this subject. And it was because I've been taking magnesium for a long time, and surprisingly, and I didn't know why, it ended up diminishing my anxiety and depressive symptoms over the time. Same happened with my husband. So when Dr. Eggert asked us to do a research project, I thought like, wow, let me find out what is magnesium doing in my body that has helped me so much. But I ended up doing a research that was very hard because it involves brain circuits. So I'm going to try to be as simple as possible. Okay, how do I move this? Okay, let me do a background of depression. Depression is one of the most burdensome, as we know, illness that affect millions of people worldwide. It has a very complex histopathology, so treatments that are available sometimes may take weeks to work and often fail to ameliorate symptoms completely. Due to its high prevalence, the study have been focused on various neurobiological biomarkers specific for depression to be able to find more suitable treatment options. Of those biomarkers, magnesium appeared to be one that plays an important role in patients with depression. So magnesium that we take for granted, well, it is one of the most important elements in the body, is the second most predominant cation in the body, involved in more or approximately 300 enzymatic reactions. And of its vital roles includes modulating brain biochemistry, intracellular transmission, myelination process, synapse formations and maintenance. Therefore, magnesium was very important to optimize neural functioning, a thing that I didn't know. So, well, here, as I said before, depression's physiopathology is extremely wide. That's what I want to show with this scheme, that it's caused by a very and a wide factors, but mainly it's caused by disturbance or dysregulation in noradrenergic neurotransmission, especially in serotonergic dysregulation. So how magnesium concentration is correlated with depression? Well, there were three mainly things that I found in few papers that I read. And first, it seems that when it's low magnesium concentrations inside the cell and high calcium and glutamate, it produces a dysregulation that may contribute, dysregulation in the brain synaptic function that may contribute with depression. So when magnesium concentrations are increased, it may regulate calcium influx into the cell and prevent dysregulation. Also may contribute providing neuroprotective properties, proliferation of neural stem cells and the dendritic outgrowth. The second factor that I found that magnesium influence in depression is that it has been found that patients suffering from depression have a HPA axis, that is the limbic hypothalamic pituitary adrenal axis, dysregulated, so it's not functioning properly in patients with depression. So magnesium also influences hormonal secretion by reducing the release of ACTH, adrenocorticotropic hormone. So apparently, without magnesium deficiency, so increasing the concentrations of magnesium inside the cell may contribute that the HPA axis functions properly. The third one, let me see, let me close this. Okay, that is the most important, and it was the hardest to understand, is that magnesium seems to be involved in the blockage of the NMDAR receptor, the same receptor that targets the fast-acting antidepressant ketamine, and also an NMDAR antagonist as well. I don't know if you remember in class when we started studying central nervous system, how doctor said that ketamine bind an NMDA receptor, and it's used in patients like as a last resource when patients have chronic deep depression and are almost suicidal. Well, apparently magnesium also binds to this receptor, so it may have antidepressant properties. Other components of glutamatergic transmission, so such as AMP receptor, appear to be modulated by magnesium as well, providing antidepressant actions. Well, this is a graph that I found in one of the papers that I read, made by Emily Tarleton and her group. And she made a clinical study, one of the few that I found, with 212 subjects. And they provide supplements of magnesium and made a health questionnaire 9 called PHQ 9 questionnaire that shows how severe are depressive symptoms in patients. So the red ones shows baseline, and the yellow ones shows the results when patients took magnesium. So we could observe that it was diminished compared to baseline in the symptoms after magnesium intake. And the blue were control. The middle line is the media. So the median were sometimes similar, but generally the orange ones or the yellow ones were lower than the control and then the baseline that were without any magnesium intake. So as a conclusion, I can say that some studies have shown that there is an inverse correlation between magnesium concentration and depression symptoms. So apparently, it has been shown that magnesium deficiency is present in patients that have depression. So for instance, increasing magnesium intake may improve depressive symptoms. However, there will be further studies to better understand how magnesium may be used as a treatment to aid in depression. What happened when I was doing this paper is that I found a lot of resources, but most of the studies have been made on rats and few studies have been made on humans. Also, because of how different humans are and all the variables that have to control, it has been difficult to keep going forward doing studies with supplements of magnesium. But it is clear that it has a correlation with depression. So I assume that they will make more studies to find an alternative treatment that I can aid patients that have from this burning disease. Thank you.
Good job.
Oh my God.
Anybody have any questions for Maria?
I have a question. How did they measure depression in rats?
How they did, let me remember. I don't remember how they measure. I don't know if it's, they studied their behavior, I assume. The behavior in rats.
Yeah, a lot of times it has to do with like, exploratory behavior or this or that. Do they just seem like they're just bummed hanging out in the corner, or are they like checking things out, and how active are they?
And they cannot, sorry to interrupt, but like do their mazes as the same rate or have like a memory impairment. But yes, it's about behavior. With humans, it's with this questionnaire and other questionnaires. Apparently this questionnaire is a little updated, but it's the one that I read that it's commonly used in the papers. The papers were amazing because I found that there is a correlation, but the explanation when they gave it, it was very hard to understand for me. And they all conclude, all the papers that I read, that they will be needed further studies. But apparently there is a correlation between magnesium concentrations, intracellular concentrations, and depressive symptoms.
Right. And that's one of the problems about going into primary literature is it gets very, very specific into the details of that level of expertise. So like, even if I'm reading an article on some area I'm not that familiar with, I'm like, what's going on here? And you know, you have to. So it's yeah, it's that's normal. It's normal when you get into the primary research. It usually is much more difficult to just absorb it without having more specific knowledge about the field. So that's
Well, thank you. Yes, when I had a slide that I want to show it because my brother advised me to, but it was from circuits, and I'm like, guy, this looks like a plate of pasta. My friends won't understand. I don't understand my yeah, but apparently I hope it will because it has helped me. So that's why I wanted to share with you, and I hope that in the future can be shared with more people because it has helped me a lot.
yeah, cool. And yeah, the other like another major like the one that Emily was saying, the BDI, that's like the Beck Depression Index. That's another very common way to look at evaluate depression in these studies. There's a few different scales that are kind of pretty well validated and people keep using. All right, so thank you.
Thank you.
All right, so we could. I'm looking here, so we could do like a. So
I
Sarah,
could go.
oh, who's that?
I could go.
Who is
Steven.
that? Stephen. Okay. Oh, Stephen will be a nice riff off of
Yeah,
this because
exactly.
he doesn't have to do as much background on depression here.
No.
So,
I'll give a little bit of background.
all right, so here we go. Let's welcome Stephen Beck to our presentation here.
Get ready.
Ready to go.
Desktop. There. No. Okay.
Again, if you're having trouble,
yeah, just one sec. Well, yeah, I think. Yeah, could you operate? I can't. It's locked on my end.
interesting. All right, so let me get it up and running, and I will. All right, so you just have to tell me, when to go.
Yeah. When to go? Okay. So let's just start with slide 1.
It's should be coming up right now.
There it is. All righty. So this segues perfectly into my topic, which was the use of SSRIs for treatment of major depression. It was interesting, there's going to be a little bit of crossover between our presentations, but I'm going to just go ahead with it. All right, so major depressive disorder or major depression is a common and serious condition. It's chronic, recurring, and life-threatening. 3 million people are estimated to suffer from depression worldwide. This is almost 5% of the world population. In the United States, about 16% of the population suffers from depression. It is estimated that 20% of women and 15% of men will suffer at least one episode of depression in their lifetime. Common symptoms include depressed mood, fatigue, thoughts of worthlessness, inappropriate guilt, hopelessness, and sleep pattern problems. For a diagnosis of depression to be made, depressive symptoms must persist for a minimum of 2 weeks. If you go to slide 3.
Okay. Hold on. This one?
One more. There we go. Perfect. One of the main treatments for depression is therapy involving selective serotonin reuptake inhibitors, or SSRIs. SSRIs increase serotonin in the brain. Serotonin is a neurotransmitter that influences memory, learning, sleep, pain, and mood. It is thought that SSRIs work by inhibiting the function of serotonin transporters. Serotonin transporters are located on presynaptic neurons. These serotonin transporters are integral membrane proteins. They are responsible for ending the action of serotonin in the synaptic cleft. They transport serotonin back from the cleft into the presynaptic terminals. Essentially, they stop the serotonin signal in the cleft. SSRIs bind to serotonin transporters on presynaptic neurons and block transport of serotonin into these neurons. The binding of SSRIs to serotonin transporters prevents serotonin reuptake into the presynaptic neuron and serotonin levels in the synaptic cleft between neurons rises. You go to the next slide. Okay, I think one back. Okay, here we go. Perfect. Nope, one more. Yeah, there. Perfect. It should be noted that it takes a few weeks for there to be any beneficial effects from taking SSRIs. Although after one begins taking an SSRI, serotonin levels in the synapses increases almost immediately. However, it takes a few weeks of continuous SSRI treatment for there to be any positive effects from taking the SSRI. This shows that in regards to SSRI treatment, there's more going on than just an increase in serotonin levels at the synaptic cleft. It is thought that the main beneficial effects from SSRIs actually occur due to downregulation of autoreceptors. Autoreceptors are located on the somatic dendritic regions of presynaptic neurons. They're actually inhibitory. Where autoreceptors bind to serotonin, they decrease the firing rate of the presynaptic neuron, which decreases serotonin levels in the cleft. As mentioned, SSRIs increase serotonin levels in the brain. Due to these increased levels, the number of autoreceptors on presynaptic neurons is reduced. This is known as downregulation. This downregulation takes a few weeks after medication is initiated. After a few weeks of SSRI treatment, there are less autoreceptors, so the presynaptic neuron is disinhibited and the firing rate is increased, which increases serotonin levels in the cleft. Thus, since downregulation of autoreceptors takes a few weeks, it takes time for SSRIs to result in any benefits for the patient. Okay, next slide. Thus, SSRIs increase levels of serotonin in the synapses of the brain. In the past, it was thought that the reason SSRIs were beneficial was simply due to the fact that they increase serotonin levels in the brain. This was known as the serotonin hypothesis. More recently, many researchers have argued that this model of how SSRIs function is overly simplistic. They contend that the beneficial effects of SSRIs are not simply due to increases in serotonin. Instead, there are other factors at play. A more current hypothesis is that SSRIs increase neurogenesis and that this is why they are beneficial. It has been found that in patients with depression, there's often a reduction in the number and density of neurons in the hippocampus. Also, brain imaging studies show a reduction in the volume of the prefrontal cortex in depressed subjects. Indeed, depression can result in atrophy of the brain. It has also been shown that people with depression have relatively low levels of brain-derived neurotrophic factor, or BDNF. BDNF is a protein that has an important role in maintaining the integrity of synapses. It is required in the central nervous system for neurogenesis to occur. It has been shown that depression can reduce levels of BDNF in the brain. Thus, patients with depression often have brain atrophy and decreased levels of BDNF. Next slide. It is thought that decreased BDNF levels leads to brain atrophy and less neurogenesis, and this then results in symptoms of depression. Many researchers now argue that SSRIs actually work by increasing levels of BDNF in the brain. A postmortem study found that subjects receiving antidepressant treatment had increased levels of BDNF when compared with depressed subjects not receiving antidepressant treatment. By increasing BDNF, SSRIs stimulate neurogenesis and new synapse formation. Increased levels of BDNF reverse the atrophy of the brain caused by depression, and this decreases symptoms of depression. Next slide. So do SSRIs actually work? Well, the evidence seems to indicate that they do work, but not very well. One study by Barboui et al. Involved a review of existing research. The researchers analyzed trials which compared the efficacies of SSRIs and placebos in treating depression. The review involved 150 randomized controlled trials and over 16,000 subjects. In the trials, subjects were either given placebo or an SSRI. The Hamilton Depression Rating Scale, or HDRS, was used to assess subjects' depression levels at baseline and after treatment. HDRS is a scale that is administered by a clinician to assess depression symptoms. A score below 7 is normal, and a score greater than 17 indicates moderate to severe depression. For the study conducted by Barboui et al., response to intervention, which was the placebo or the SRI, was defined as a 50% reduction in symptoms on the HDRS scale. The researchers found that 51% of subjects responded to an SSRI, while 31% responded to placebo. This review shows that SSRIs are modestly effective, more effective than a placebo. Next slide. Another meta-analysis showed that the average response rate to SSRIs was 54%. In contrast, 37% of subjects responded to placebo. There was only a 17% difference between treatment versus placebo in this study. Again, this shows that SSRIs have small benefits. Okay, next slide. Though much of the research suggests modest benefits from SSRIs, some researchers argue that SSRIs are ineffective at treating depression. Kirsch et al. Looked at data from SSRI trials submitted to the US Food and Drug Administration. They found that on average, SSRIs improved the scores of patients on the HDRS scale by only 1.8 points more than the placebo. According to the UK National Institute for Health and Clinical Excellence, a significant clinical benefit for antidepressants is a drug placebo difference in the improvement of HDRS scores of 3 points. Thus, since the meta-analysis performed by Kirsch et al. Found that SSRIs improved the average HDRS score of subjects by only 1.8 points more than the placebo, it can be argued that SSRIs do not offer clinical significant benefits to depressed patients. Okay, last slide. In summary, most of the research shows that SSRIs offer only modest benefits to depression sufferers. It was mentioned that many experts now believe that SSRIs work by stimulating neurogenesis. Hopefully, in the future, medications specifically tailored to promote neurogenesis might be developed to offer greater benefits to patients suffering from depression. It's interesting to note that ketamine, which was talked about in a previous presentation, has recently been shown to be effective at treating depression. Instead of increasing serotonin levels, ketamine increases BDNF, which leads to neurogenesis. Treatment with ketamine leads to formation of new neurons. Unfortunately, ketamine does have a high potential for abuse, so researchers are looking for other agents that might increase levels of BDNF and rates of neurogenesis. That's it.
Okay, thank you.
Super cool.
And so does anyone have questions for Stephen here?
Stephen, yours would have been perfect as an introduction to mine.
Thank you.
Amazing. Yeah.
Right, and I mean, this also brings up the thing about trying to look at, the average, what's the average effect of something versus, looking at any individual. Kind of when we think back to the blood pressure lab even, like how individual responses are. Like even if on average, these drugs might not have that dramatic of effect, I think they probably would not be in the pharmacopoeia if like for certain people they are very effective. So it's kind of an interesting thing. You have to be careful looking at a big average versus looking at like what is it for one or another person as well. So that's any other last questions for Stephen here? So let's do one more and then we'll take a break and then do some more. So let's for our next our last presentation before the break. Does anybody want to go?
I can go.
All right, so we'll have Sari, and can you share, or would you like me to do yours?
I will try to share. Let's see.
All right, so
It says I can't. Well, oh, but I think Stephen, are you still sharing?
here I just stopped. I kicked them out, so you should be able to share now.
Great.
Oh, there we go.
Okay, cool. And then how do you do slideshow?
All right.
Okay. Well, this is exciting because it goes very much off of Emily's too. So that was great. I'm excited. Yours was amazing and provided a really great, preemptive around probiotics, and mine focuses on the benefits of breast milk on the developing infant's immune system. And I want to preface that there are so many amazing benefits of breast milk that are far beyond just the impact on the immune system, but I wanted to go specific. But there's huge benefits for the developing infant, for the birthing parent, preventing obesity, food regulation, depression, regulation of weight for the birthing parent. So I hope this strikes you to go investigate breastfeeding or, other ways of talking about it as chest feeding or body feeding or other ways. But for this presentation, I'll just say breastfeeding and breast milk. Okay. So partly what's so interesting about breast milk is similar to what Emily was talking about with the probiotics, that there's these bioactive properties of breast milk that provide innumerable benefits for the developing infant. And so on the right, you can see, for formula does a great job if that's what's available. And again, this is contingent if breast milk is available. And I know that people have such different circumstances of availability of breast milk, but formula will do a good job growing your baby. And it has the minerals, it has the vitamins, and it will make your baby increase weight. What's really exciting about what's in breast milk, it has all these bioactive properties like antibodies, hormones, antiviral properties, antifungal properties, protection of infection, enzymes that basically provide scaffolding for the baby's immune system for both short-term and long-term benefits. And also been linked to preventing food allergies specifically as well. And it's currently the preeminent recommendation for optimal choice for nutrition for infants from birth until at least 6 months of age. And there's a lot of variety of, recommendations of when to stop. But in my understanding really is as long as the baby wants to continue eating breast milk, then that can continue. So there's not like a limit on it. Oops. Okay. Oh, shoot. Can you not see on the left?
Yeah, it's cut off.
Well, okay. I might go back to the, okay, sorry, not ideal. Can you see it now?
Yeah.
Okay, I'll go back to the presentation afterwards. But so narrowing in on the impact of breast milk on developing infants' immune system, it strengthens host defense mechanisms against infectious agents and pathogens. And it has those bioactive health factors that I just mentioned before, and that Emily talked about quite a bit too, of reducing incidence of disease, promoting gastrointestinal immunoregulatory functioning, and GI mucosal maturation, decreasing prevalence of infection, really increasing the diversity of the gut microbiota, especially in that initial scaffolding when the baby is first breastfeeding, specifically with colostrum, which is the first, sort of this thick breast milk that comes right out of the breast at the beginning. And there's this huge movement right now too, after the 60s, basically of ensuring that skin to skin and breastfeeding happens right after the birth so that this baby can have this big immune boost, basically. And like I mentioned before, it offers immune protection beyond the termination of lactation too. Okay, I'll go back to, slideshow. Oop. This more, instead of going over each one, is just to show you there's huge amounts of statistics, that different studies have conducted around the benefits specifically on breast milk on the infant's immune system. Some of them 64% reduction in the incidence of non-specific GI infections in infants, lowering diarrhea and protection against symptomatic, asymptomatic giardia, and prolonged diarrhea, which is a huge cause of mortality in infants. So the link also there of preventing mortality through breastfeeding. Non-breastfed children have almost 15 times increased mortality rate from pneumonia compared to children who were EBF, just means exclusively breastfed. And there's also a lot of studies that say even a little bit of breast milk does this immune scaffolding. So, these studies, there's a lot around exclusively breastfed, but also the benefits of partially breastfed too, if that's more available for the birthing parent. So for example, infants fed any amount of breast milk have shown a 23% lower risk of developing acute otitis media for at least 3 months and reduced 50 to 60% when exclusively breastfed for 6 months. So again, this page, instead of going over each one, is more to say, there's all these studies that are really showing the benefits on the immune system. So partly of it, what I really focused on in my paper and what Emily did a great job talking about was, how baby, and what Professor talked about earlier, of the passive, basically getting microbiota and protection from breastfeeding. And so the baby inherits all these bioactive health factors and microbiota, which is just microorganisms or the probiotics that Emily talked about. And initially it completely colonizes the infant's gut. And it's shown in all these studies too that babies that have been breastfed have huge dominance in their initial gut of bifidobacterium. And I specifically focus on bifidobacterium longus and the substrate of infantis, I believe it's pronounced. And these leads to increased immune functioning, impact against allergy development, and infection. And so what is bifidobacterium? So as Emily talked about too, it's a probiotic, it's naturally occurring, it's helpful between host and for the bacteria too. It is found in the GI tract and also vagina of animals. This is bifidobacterium longus I'm talking about. And it basically inhibits other pathogenic bacteria. Dr. E talked earlier about the production, the metabolism that produces acid, which lowers the pH. And that does twofold of protecting against other, pathogens from invading, as well as creating a dominance in the gut, which really promotes, all this multiplicity of benefits on the infant. And so specifically, like I mentioned, bifidobacterium longum promotes immunoregulatory factors that affect both local and systemic levels. It encourages gut homeostasis, regulation of both the innate and adaptive immune responses, which we just learned about today, so very on point, and decreases inflammation and can compete against other bacteria. And so there was this one study that I thought was really interesting that dove into the subspecies of B. longus, which is B. infantis, and that in breast milk, it contains these high levels of free sugars called human milk oligosaccharides, and it makes up a third largest component of breast milk, but it can't be digested by other enzymes. And this is where B. infantis comes in, where it can utilize those HMOs. So that's what's really exciting, was finding that babies who were breastfed had really high levels of B. infantis. And when they metabolize these sugars on HMOs, they created this metabolite, which is just a production of metabolism, and created these things called ILA, which reduces, as Emily talked about, the LPS, which is lipopolysaccharides, which is a signal of inflammation. And so what's exciting about it is that breastfed infants that have these high levels of B. infantis can basically use these HMOs to create a metabolite that lowers inflammation. And that's one of the reasons it's so beneficial for immunoregulatory responses. So I just talked about that here, about ILA metabolites. This basically just goes over it in more detail again. And there was just a bunch of studies, proving the dominance of ILA levels in breastfed babies, and that how that basically lowers the inflammation responses. Lastly, the effect on allergies too is the breast milk is the current recommendation to prevent allergic diseases, including food allergies. And it offers protective mechanisms against cow milk allergies in early childhood and among high-risk infants too. And there's shown lower incidence of immune-related conditions. And on the offside of that, infants that were not breastfed showed lower incidences of bifidobacterium colonization in their gut and more increased tendencies for allergy development and a myriad of other diseases. And this kind of shows you in a visual form at the bottom there, the difference of how bifidobacterium longus alleviates allergic disorders. That's on the right. So my closing thoughts are that breastfeeding is amazing and there's so much more to it, but specifically on the immune system, the impact of it is so profound, and that it's exciting to see all the ways that people are learning about immunoregulatory mechanisms and how these specific bacteria that Emily, presented in the beginning can be really helpful in the early gut colonization of infants to then provide immunoscaffolding for the rest of their life, and especially in that first year of life. So thank you for listening. Appreciate it. Here for questions.
All right, that was awesome. Are there any questions for Sari?
I was wondering if in your research of like relating to allergies, if you found that if like a parent had an allergy, if that was then passed on to their kid.
So that was one of the articles I read, was that even in parents that have presenting allergies, if they breastfed their babies, they wouldn't develop the same allergy. But that was one article. So I can't speak to, but it's a good question. I'm not sure.
Super cool.
Did you come across any data about donated breast milk?
So not necessarily in this research, but I teach online breastfeeding classes for homeless pregnant people in San Francisco. And so when I talked to the lactation consultant, she was saying that, it's an amazing alternative, although really expensive. So depending on your position, but that the problem potentially is that the antiviral properties and antifungal properties might not be as profound because it's being stored. And even though like if it's in the fridge or in the freezer, it can maintain those, those bioactive properties, but I'm not sure in those like how long they're being stored for. So it'd be a really good question to know the parameters on that and seeing if those are still really like alive, if that makes sense.
Interesting. Thank you.
Cool. Yeah, and the whole question about kind of back to the immune system and this microbiota, it's more and more clear that your immune system does not actually develop quite right if you don't have the appropriate bacteria around, and things can get over, too twitchy, where you start having the immune system starting to cause inflammation when it shouldn't, because it's just it's not used to seeing the normal stuff around, and just like, oh, I don't got to worry about that, and it's like, oh, something I gotta like attack. So like, yeah, having the appropriate like bacteria around during development really seems to play a role in having your immune system kind of developing well.
And it is exciting in this moment in time too, because like with the medicalization of birth, there was this huge movement by doctors of sterilization, and sort of what Emily was talking about too in this moment, where it's like saying, oh, breast milk's dirty, breast milk's bad. And so then the whole culture on breastfeeding got diminished in the States I'm talking about. And formula was kind of partnered with the pharmaceuticals and the hospital industry. And so that's the promotion of formula. And so only in the past like 30 years has there been an influx of trying to reimagine the culture of breastfeeding and institute that. And now colostrum and skin to skin, like I was talking about, is coming back, but it's, definitely slower, but people are finding like, oh my, there's these huge benefits of breast milk, and it's free.
Cool. All right. And it does, I think it's also worth, just because some women have trouble doing that, like kids who don't have breast milk grow up just fine. So you don't want to make somebody feel like they've been a bad mother because they can't nurse.
And that's why I had my asterisk that there's so many different circumstances. People are coming from all different places, but
I do want to just add that into there. It does seem to be a benefit, but if it's not not possible, it doesn't mean like the kid is not going to be okay. So so let's take a little break, and then we'll have time for at least a couple of more. All right, it is noon, and I think we had Morgan in the queue here. So, Mr. Morgan Rux.
All right, let me grab my stuff real quick.
All right, so let's welcome Morgan to our presentation rotation here.
All right. How's everyone doing today? So my research paper was on the mammalian dive reflex or mammalian dive response, kind of focusing on the human aspect of it. So yeah, let me try to move this little bar here. Sorry. Okay, so the mammalian dive response is a protective, physiologically multifaceted response to apnea and cold water submersion that occurs in all mammals, including humans. it's like a broader response than that. It's found in pretty much every vertebrae that's studied. It's a really old, highly conserved response. So the parts of it, the parts of the dive response is apnea, just not breathing, bradycardia, which is your heart slowing down, peripheral vasoconstriction, which is the arteries and veins and capillaries in your limbs and your peripheral squeezing down. And then the last part is splenic contraction, which is your spleen squeezes. Yeah. So I'm gonna kind of go over the parts of it. This picture right here is it's a Japanese pearl diver, the Alma people. They've been diving for food and pearls for a long, long time. They're really cool. Interestingly, if you look at traditional cultures that do free diving, it's typically the women that do it, not the men, because they have more subcutaneous fat. You know, they have like equal distribution of fat over the whole bodies versus us guys, we just get beer bellies. So they are a lot more thermal tolerant because in the days before wetsuits, you know, water's cold. But anyway, what initiates your dive response is cold water is detected on your face by thermoreceptors, and this information is relayed to your brain on the trigeminal nerve. And then the carotid body chemoreceptors they monitor your CO2 levels and your oxygen levels in your central circulation, and they notice rising carbon dioxide levels and decreasing oxygen levels, and they also initiate the response. And then the actual part of the brainstem that seems to be implicated in initiating that response is in your medulla oblongata, the dorsal horns, and then a couple other little parts in there. The science that kind of shows this is that you can use lidocaine or bupivacaine to numb up somebody's trigeminal nerve, and then if you make their face cold, they won't have a strong dive response. That's the first part. And then for the carotid body, they compared people that had their carotid bodies actually taken out of them, because that used to be an old-timey treatment for asthma, severe asthma, which is pretty intense. And those people with cold water and breath hold wouldn't have the bradycardia that you see in normal people. All right, so upon breath hold and cold water to the face, the vagus nerve innervates your heart, right? And that releases acetylcholine, which interacts with M2 muscarinic receptors. We go back to our studies on the parasympathetic nervous system, which that causes a decrease in heart rate. The next part of the dive response is peripheral vasoconstriction. So the dive response is kind of trippy. It's not just parasympathetic, it's both parasympathetic and sympathetic. So at the same time as you have the bradycardia happening through the parasympathetic division of your autonomic nervous system, the sympathetic nervous system is innervating arteries and veins in the periphery, which have vasoconstriction. This is a couple of things. One, it shunts blood from the muscles, which are tolerant to not getting oxygen. And it also increases your peripheral vascular resistance, which means that you maintain your blood pressure even in the face of the bradycardia. If you remember our little equation on, blood pressure is determined by like heart rate, stroke volume, and peripheral vasoconstriction or vascular resistance. I'm sorry. And what's interesting too is typically at the capillary beds, if you have increasing carbon dioxide levels and decreasing oxygen levels, local control will open up those blood vessels again to clear those wastes. but this dive response is able to override that local control. There's variation in genetics that we see that affects how strong that is. I'm going to go over a couple studies that kind of demonstrate that. And so the last part of the dive response is splenic contraction. And so this was first noticed in marine mammals. The first one was like the Weddell seal, which is a big seal that lives near Antarctica. And like it has a spleen that will hold 20 liters of packed blood cells that have a hematocrit of like almost 90%. So it's like really concentrated, oxygenated blood. And they had some really amazing studies where they're able to measure their hematocrit measure their hematocrit in real time as they're diving, and their actual hematocrit will go up through the course of their dive because they're releasing that fresh blood into the central circulation. And for a long time, we didn't really think that was a thing that happened in people. We have kind of a small spleen. But then they studied the Alma people in Japan, and then there's another tribe in island tribe off the shore of Korea that is a traditional freedive culture. And they used CT and ultrasound, and they found that their spleen is like much larger than the average spleen, which is very cool. So this is a cool study. And this is on this little picture here is a little plug for underwater rugby. If you like holding your breath, it's like the funnest sport of all time. That's my friend Jackie holding the ball there, about to score on somebody. So this study in 2008 compared the Baju spleen size, which is the Korean traditional freediving culture. And what they did is they compared their spleen size of the people within the tribe to another group of people that lived adjacent but not on the ocean. They lived about 25 kilometers away. And they found that their spleen was much bigger. And then they did genetic studies of them and then other people in the region, and they found some pretty interesting things. First was a single nucleotide polymorphism on this gene, FAM17.8B. And, genetics is a little bit above my pay grade, but basically, they looked at these single nucleotide polymorphisms, and they found a bunch that were different, but I'm going to go over 3 that were interesting. One is this gene, which encodes a protein that interacts and may allosterically regulate carboxyl anhydrase, which is really important to your CO2 buffering system. And so they may be able to maintain a more neutral pH in the face of more CO2 in their blood. And then the second one was involved in the regulation of glutamate, which is the most abundant neurotransmitter in the nervous system and is important, especially in the signaling of that peripheral vasoconstriction. And then the third thing they found was this other genetic variation on a words I write but don't say often. A phosphorodiesterase that is involved in the regulation and contraction of the smooth muscle that surrounds the spleen and blood vessels, and is involved in your cyclic AMP, cyclic GMP signaling. So this research found a possible genetic mechanism for the variation of the dive response seen in people. All right, second study I'm going to go over. This one was cool. They took a decent sized population of people, and they used PCR assays to look for a few different genetic polymorphisms in the renin-angiotensin and the kinin-bradykinin genes. In class, we went over the renin-angiotensin system, if you remember that. It's involved in peripheral vasoconstriction and blood pressure control. And so they looked for that polymorphism. They found it in some part of that population, and then they kind of predicted how strong their peripheral vasoconstriction would be in response to the dive response and apnea. And then so they stimulated the dive response using cold water and holding the breath. And the study showed that people that were homozygous in these places had much stronger peripheral vasoconstriction. And so the subjects that are homozygous in the DC ACE genotype, which is involved in your ACE signaling pathway, had 2 times the levels of angiotensin converting enzyme compared to the other people. So this study shows another genetic link to control of this dive response in humans. And so yeah, that's what I got. Anybody have any questions?
Any questions for Morgan?
Morgan, it was super interesting. I didn't know that diving in cold water could have all these benefits and epigenetics. It is.
So that's so when they looked at those genetic polymorphisms that were specific in the diving, what were they, Bajows?
Yeah.
Are these populations really isolated? So they don't move around much or intermix?
Yeah, sorry. Yeah, so they live in an island. So I don't know exactly how isolated they are.
Okay, yeah, that's trippy that you have people who are kind of, this little subset really kind of evolved to be optimized for diving in cold water like this.
Yeah, and I mean, there's the nature nurture dichotomy, right? You have the genes you're given, but you can definitely change your effects. Some of the research I was looking into was comparing free divers or people that do breath hold diving and people that don't, and people that do have much bigger spleens.
Right. Yeah, that part I could see being more of the nurture part, but just the distribution of different alleles in a population, that's intense.
Yeah.
Cool.
All right.
So I guess we have time for one last one, and I think Marissa Morris asked early on, so I think we'll finish up our Tuesday hearing from Marissa Morris, and we need to stop sharing.
Yeah, I'm not sure how to do that on my end. Are you able to?
I'll do it for you.
Thank you so much. Okay.
All right, so now let's have Marissa Morris bring us to the end of the class here.
All right, so I did my research on migraines and the risk of CVAs and CVD, which is cerebrovascular accidents and cardiovascular disease, so also known as strokes. So basically, with migraines closely mimicking stroke symptoms, I sought out to find the relationship between migraines and the risk of CVA and CVD. This actually kind of piqued my interest when I was doing my schooling for EMT. We were going over the cardiovascular system, and our teachers were going over the stroke symptoms. And when he was going over that, I noticed how closely stroke symptoms mimicked my own migraines. And I've suffered from migraines since 3rd grade. I'm now 35. So it's been a long time of me dealing with migraines. So
when I took these courses and they were mimicking strokes, I was like, oh, great. So am I having a stroke or am I actually having a migraine? So, when we had this assignment, I just really wanted to dive in to see, if there was any research that actually linked them. So what is a migraine? Basically, migraines are headaches that are ranged from moderate to severe pain. They can be throbbing, they can be pulsing. They typically happen on one side of your head, and they are seen as disorders of the brain. There's many factors that can cause them. It could be genes, it could be your environment, internal factors, which plays into vascular issues. And there are two types of migraines that are commonly talked about. One is migraine with aura, and the other one is migraine without aura. And unfortunately, women are more likely to suffer from migraines than men. So as we can see here, you can kind of see the male and female, male being in blue, females being in red. You can see how much more women suffer from migraines than men. So what really causes migraines is still unknown. There are some common triggers such as hormones, stress, caffeine, skipped meals, tobacco, medications, processed and cured meats. So like your salamis, basically any kind of Italian meats, pepperoni, even lunch meats. If you actually look at the ingredients of your lunch meats, they have sodium nitrate and nitrite. Those are very common to trigger migraines. Chocolate and certain fruits. I myself, my migraine started with strawberries. And my doctor said that is actually a very common trigger for migraine sufferers is actually strawberries. I guess it's the citrus or something in the strawberries that is a common trigger for them. And then also the nitrates and nitrites in like the deli meats and anything like that is also common triggers. So migraine with aura is the most common type of migraine that people experience, with 1 out of 3 people experiencing it. It's often characterized by reversible focal neurological symptoms. They can develop either before or during a migraine, and they don't last for more than an hour. So basically those include sensitivity to light or sound, nausea, vomiting. You can experience tingling and or numbness in like your fingers, your feet, entire body. Also difficulty speaking and vision loss. So looking at those symptoms, those also very likely mimic strokes, like people lose feeling in one side of their body. They have difficulty speaking, even though they don't perceive it as it, but other people are like, what are you talking about? So these symptoms really closely mimic a stroke. So you can see that there's 2 out of 3, in a study of 2 out of 3 people, women are most likely to have migraines. And the frequency of your migraines increases your risk of a CVA. Another reason why women are more likely to experience them is, obviously hormonal. We often take, birth control pills. Your risk increases with cigarette smoking as well. And another thing that they've found with migraines and relations to CVAs is something called cortical spreading depression. A lot of studies have actually found this common link, and a lot of the studies I looked at mentioned it. So what exactly is this? It's a mechanism that plays a role in the development of strokes and brain injuries. It basically changes the permeability of the blood-brain barrier, and it's associated with significant ionic changes. So basically, it just increases the blood flow, and then the prolonged reduction in cerebral blood perfusion, and it just decreases the vascular responsiveness. So I kind of have this little thing. So this is over time, and it kind of just spreads. So with migraines and cardiovascular disease, basically the factors are pretty generic in all kind of cardiovascular diseases, the increased lipid levels, diabetes, elevated blood pressure, and then history of early onset vascular disease. What we have also seen is that some of the common medications that are used to treat migraines can actually increase your risk for cardiovascular disease because they constrict the coronary arteries. So like Imitrex, I know is very popular. I was actually prescribed that when I was suffering from migraines a lot. And so, kind of just looking at all these studies and how there's certain factors that could contribute to higher incidences of cardiovascular disease and CVAs was kind of shocking. So basically, we still don't know the exact causes of migraines. We only treat the pain. As of right now, they are not curable. There's no direct causation either way. We can't really see if migraines cause CVAs or CVD and vice versa. But we are noticing that people who experience migraines with aura are twice as likely to experience a CVA. So with that, I think that doctors should really consider looking more deeply into someone's history with their risk of having a CVA. It's just, we just can't find enough solid research which causes what. And obviously, genetics plays a huge role in that too. But like I said, they haven't really found, concrete evidence as of what causes what. So it could be migraines that are contributing to more CVA, CVD, or it could be vice versa. We just have to do more studies on it and see what we can find.
All right, thank you. Do we have any questions for Marissa?
Just out of curiosity, did you happen to find anything on just alternative treatments for migraines?
I didn't. It was just mostly talking about, just medications and stuff like that, but nothing like therapeutic or anything like that. Obviously stress can bring them on as well. So, I mean, it's just kind of finding your niche. Also too, exercise can bring them on too. It's just, it's so varied across everyone that, it's like they can't really pinpoint what exactly causes and what can work. It's just like with everything else, it's kind of on an individual base. I know for myself, a lot of people have experienced really good results from Imitrex. It made mine worse. And, I've discovered as I got older that my migraines went from hormonal to now when I get them, I'm dehydrated. So it's just kind of this whole like span of, what works for people.
Right. I mean, there's actually, there's work on psychedelics like psilocybin in addressing migraines or certain types of
I
migraines.
saw that as well. There's a guy, cluster headaches, I think
Yeah,
they call them, and
cluster
he was
headaches,
taking
exactly.
magic mushrooms for him. Yeah.
Yeah, it's kind of interesting. All righty.